From Microbe TV, this is a special immune booster recorded during the American Association of Immunologists conference in Chicago, May 2024, starring Cindy Leifer and Brianne Barker, and featuring Mark Shlomchik from the University of Pittsburgh.
Welcome.
Thank you.
So we just kind of wanted to start by hearing a little bit about you. So when did you first become interested in science and sort of your path through science to where you are now?
Oh, I had a weird path into this. I will say that I was always interested in science since I was a little kid. But I always thought I was going to medical school. And in college, I got really into developmental biology, and I started even taking graduate courses. And then I just applied to medical school, went there. And I found out in the first year of medical school, I was getting really depressed. I really didn't like medical school. In fact, maybe I even hated it. I don't know what was really going on. But I could do it. It was all about memorization. But I realized it wasn't what I thought and what I really wanted to do. And most of my classmates were trying to, basically, they didn't understand why we were learning all this basic science. They just wanted somebody to tell them what to do. And I was sort of feeling differently. And we had an immunology course in that first year that was taught by Michael Cancro, well-known B-cell biologist. Yes. He was a new assistant professor at the time. Oh, wow. And he was he was teaching all the lectures, and it was great. He was teaching it like a graduate course. And he was also my small group preceptor. Mm-hmm. He gave a very different kind of final exam, like design an experiment, interpret the data and all that, which I really loved. And on my final exam, when he turned it back, he said, he wrote, "This is a great exam. If you ever want to do research, give me a call."
Oh, that's amazing.
And that just it did really ring a bell. Yeah. And I still remember, he wrote his phone number. I still remember what it was. No email in those days. And I decided I'm going to give him a call. And sort of the rest is sort of history after that. I was kind of scared to tell my parents that I was going to take a year off from medical school, which is how it all got started. But they were okay with that. And I went to work for him in the lab that summer after my first year of medical school. By the end of the summer, I knew I was applying to graduate school. And I started taking the graduate courses. It took me a year to cycle in, but they accepted me. And so then I was part of the MD PhD program. Okay. Except I never really did the right MD PhD program because I was so weirdly out of cycle. Anyway, long story short, Mike actually suggested that I go and work for Martin Weigert, who is a very well-known B-cell biologist in those days, a molecular immunologist. And I did. I did my PhD in his lab. It took me a bunch of years, but I really enjoyed it. Had a lot of papers and was starting to, I could see I was having an impact in the field. Yeah. And then it was really a question of, am I going to go back to medical school or not? And then I did get persuaded to go back to medical school, finished and did got my MD PhD and even did a residency in clinical pathology. And then got my first job. Went back to Martin's lab and did a short postdoc and got my first job at Yale University. A little more than 30 years ago. Yeah. And in fact, we just had the 30th anniversary. I just had a reunion of my lab, of all the people who worked in my lab, the 30th anniversary.
Oh, that must have been really interesting.
It was a lot of fun.
That sounds awesome. So what types of things were you working on in the graduate school and postdoc experience?
Well, basically, I'm still working on the same stuff.
B-cell.
I'm still, I seemingly start a project and never finish it. But I was working on really what was driving autoantibodies and autoreactive B cells. And the first thing that we found is that they were clonally expanded and that they had somatic hypermutations and they were affinity matured. And this seems all natural now, but nobody knew that at the time. Mm-hmm. So these were important papers. And that sort of changed everybody's thinking about the specificity. A lot of people thought autoimmunity was your immune system on, drugs, kind of it was just super hyperactivated, but not specifically.
But not specific. Mm-hmm.
Right. And that because your repertoire had autoantibodies in it, it would get just activated. And we really showed, no, it was very driven, just like a chronic viral response. It was not even really known that somatic mutations were where they were happening or anything like that. So that got me into understanding. Eventually, I wanted to understand how a particular autoreactive B cell was regulated, like when it developed. So we made some B cell receptor transgenic mice to study that. That's what I worked on in my postdoc. We're still using them.
Yeah.
I'm still using all the models. And still the fundamental questions in our lab are really what drives, why do you get autoimmunity in the B cell compartment? And, eventually that led to toll-like receptors and a lot of other things. But I feel like I'm still sort of working in a large way on the same things that I was interested in the first place, which shows you a problem can evolve and you can solve parts of it, but you still always have more questions that rise up. Yeah.
Good research leads to more questions. Can you talk a little bit about how, you said you developed this model and then you're still using it now. So talking about how thinking carefully about building a really good model and then how that leads to longevity. Like, how did you think about building this model? And did you know at the time that it was really going to be able to lead to as many discoveries as
not really. I mean, we were building it, we were really interested in early self-tolerance. At the time, people were working on clonal deletion, receptor editing, and B-cell anergy, and some, other labs that were working in parallel with us, like Chris Goodnow and David Namazi, had developed these very elegant systems, but they weren't for real autoantigens. And what Martin and I decided we wanted to do was to make B-cell receptor transgenics for real self-antigens. So anti-DNA was one that he did a lot, this very famous one called 3H9. And then I worked on rheumatoid factor. Right. But they were all based on hybridomas that I had sequenced before. And we knew about them. We knew what their specificities were. We knew they had somatic mutations. And we were just interested in really, did they make it out of the bone marrow? Did they make it to the periphery? Could we put them in an autoimmune prone background and visualize them getting activated? So it sort of all led from there. I didn't know how I started working on the rheumatoid factor BCR, which we call AM14. Yep. And we didn't know what it was going to do, but it turned out to do some really interesting things. It wasn't self-tolerized in normal settings, but if you put it in an autoimmune setting, it was activated in a very particular way. And that led us to study that. And then that also led us to toll-like receptors. So AM14 stands for Anne Marshak.
Yeah. She's a good friend of mine.
Yeah, Anne is great. She just called me on the phone right as I was on my way here. So she and I were working together. I was collaborating with her when I was a graduate student, actually. And we've been working together ever since. Yeah. We still are. And she was making the hybridomas and I was sequencing them. I had figured out a method to sequence before there was PCR. So I was sequencing 2 or 3 hybridomas a week, which was a lot, back in the mid 80s, actually. Yeah. And when we published the first paper, we quadrupled the number of sequences that were in the database, the whole database. Wow. It's crazy now because, you could just do single cell RNA-seq. Yeah, you get 10,000 sequences. Yeah. But at the time, that's what we could do. But Anne gave me these hybridomas with 17-digit names. Yeah. So it's no, I'm not going to keep track of that. It's AM1, AM2, AM3. So AM14 was the 14th one. I got up to AM36, I think, in the whole thing. But AM14 had certain properties that made it good for making this BCR transgenic model that really was made by Martin Weigert, and I helped to characterize it, when I was a postdoc.
And so what are some of the discoveries that you've made with that?
Well, we learned that, first of all, we could formally show that they were activated in an antigen-driven way. Mm-hmm. So we made autoimmune-prone mice that did and didn't have the self-antigen, and nothing happened to those B cells when the self-antigen was not there. Mm-hmm. And then when we put it back in, they would get activated and proliferate. And that was because the AM14 rheumatoid factor was allotype specific. Mm-hmm. So it recognized IgHA, but not IgHB. So we were able to prove that. And then we noticed that these B cells were getting activated through the extrafollicular response. They weren't going through germinal centers at all. And that really clued me into the idea that extrafollicular responses were involved in inflammatory processes. And it's literally taken 20-something years for everyone to sort of catch up to that. But everyone thought it had to be germinal centers. And we were able to use microdissection of these extrafollicular sites to show that actually there was somatic hypermutation going on. We could build clonal trees, we could sequence the V regions and show that mutation wasn't just restricted to the germinal center. It happens, of course, in the germinal center, but it happens in these chronic extrafollicular responses. So that was another discovery. So three things that came out. One was antigen-driven. Second was extrafollicular site. And third was somatic hypermutation happening at that extrafollicular site. And we could show that there was affinity maturation and selection there. And then working with Anne, this model system eventually became our link to figuring out the toll-like receptors.
Yeah, that was what I was going to. How did you end up in that? That's how I became interested in your work. So,
yeah. So, I mean, and this is really a lot of credit goes to Anne in this. Yeah. So we were working together, and we reasoned that the AM14 B cells were probably important antigen presenting cells to break tolerance in the T cell compartment, which they are, and we're actually still working on that. And so Anne was really working on the T cell side at that time. So she was trying to figure out if she could activate T cells with the AM14 B cells, and we were feeding them these chromatin immune complexes. So rheumatoid factors are IgG specific. So if you take an IgG anti-DNA antibody or an anti-chromatin antibody, that will make a natural immune complex. There's always chromatin everywhere. Yep. And so that would cross-link the B cell receptor, would activate the AM14 B cells. We showed that in vitro and in vivo. And then we reasoned that they would bring in the proteins that were stuck on that, and then present the peptides to the T cells.
Right, yeah.
So, and then Anne wanted to find the T cells. But as is her way, she immediately noticed something weird was going on that was even maybe more interesting. And that was when she put the immune complexes with the AM14 B cells without any T cell help or anything. That was enough to get them going crazy in vitro, even better than BCR like anti-mu. And so, but if you made immune complexes that didn't have chromatin in them, nothing happened to those B cells. They were just perfectly cool. So we realized, there's got to be a receptor for, we thought, DNA there. Mm-hmm. That was before TLR9 or any of the toll-like receptors were cloned. And so we were wandering around. There were some other DNA receptors, and nothing was really working. And Anne and her student, Liz Ledbetter, who's actually here at this meeting.
Oh, she is?
Yeah. We're trying to figure this out. And then when toll-like receptor 9 was cloned, so Anne was really working already with people like Doug Gollenbach. She was very tuned into innate immunity. So she was very aware of this. She realized, that's the receptor. And so she was able to get MyD88 knockout mice, cross them onto the AM14, and that completely extinguished the whole in vitro response.
Yeah, that was an amazing
And that paper. Was great. I mean, the whole paper, all that was in it was that in vitro thing. But it was so this is a big answer. Because we really always wanted to know, how does innate immunity activate autoimmunity?
Yep. And
this was a big answer to that. And then I was working a lot with in vivo genetics. So I was the one who then picked all that up and translated it in vivo and showed that, yes, if you don't have TLR9, you lose your whole anti-chromatin ANA. And then we extended it to TLR7. That was easy to think of. All the anti-RNA antibodies were dependent on TLR7. And if you just knock out both of them, you lose your whole anti-nuclear antibody response. We were doing this in a particular mouse model, but it's been replicated in many different models. And I think it's pretty clear. And then from there, though, what we stumbled on this unexpected finding, which was that if you knock out TLR9, the disease gets worse.
Yeah, and that would have not been predicted, right?
No.
So did you?
We didn't
was it
predict
known that RNA antibodies came first? So there's a progression in the types of antibodies that you develop
it wasn't
in lupus,
really known.
But nobody really.
People think that. I don't know if that's really true or not. But we were not expecting that. It's we thought, we're going to knock out TLR9, and that's going to be the cure for the disease.
Yeah, right.
And then we'll develop drugs that will inhibit it. And this will be a way to treat lupus.
Right.
And,
easy peasy.
I remember my student coming to me and saying, there's something wrong with these TLR9 knockout mice. They're dying. I can't even get them to breed practically. And we quickly realized, this is making disease worse. But then we found out that TLR7 was the opposite. It was ameliorating disease.
Yeah.
And that was also unexpected because how these TLRs are supposed to kind of be similar to each other.
Yeah.
And that sort of set me off working on that. I'm still working on that. I talked about this at the TOL meeting. So we, I think we're coming closer to having some, at least molecular understanding, but it's not a signaling based understanding yet of why that is. But that was also replicated in many different lupus models. Whenever you knock out TLR9, you lose a repression. So something about that TLR is different. And it's got a repressive signal in it. It has an activating signal as well as a repressive signal. TLR7 just seems to have an activating signal. So the worst disease that you get when you knock out TLR9 is completely cured when you knock out TLR7 as well. In fact, the double knockout has even got less disease. And in fact, there are still companies and people working on mostly TLR7 and 8 inhibitors. And, maybe they're going to have some efficacy. We'll see. I don't know if they're good enough or if that's really the whole story. But that sort of takes me up to today. And so now, I just basically told you how starting with sequencing AM14 as a graduate student all the way to now, we're still working on that model. And it just kept evolving different questions that we had to answer. And now, we're, I think, on the verge of at least having a molecular understanding of what's different about TLR9.
Yeah, I'm really interested in that question too.
Well, I know from your heritage, you're a TLR9 person.
I mean, just the whole idea that we all thought that the cytoplasmic tails all are very similar in sequence and should just similarly signal, and that doesn't seem to be the case.
I think
you're blowing my mind here a little bit.
No, it's definitely. I think we all have to realize that we're, we do certain experiments, but the more interesting stuff turns out to be the stuff we didn't expect, right?
Right.
But I think, it's not like you just sort of try this, that, and the other thing. I mean, you have to be driving along a pathway of trying to answer what you think is an important question. That, when I talk to junior faculty about what to do or not to do, the most important thing I think is pick a really good unsolved question that's fundamental, that people would really want to know the answer to. Because I see a lot of people doing really nice science. I mean, quality controls and technical and everything like that. But it's sometimes not always focused on the most important questions. And then they wonder why this is so good. Why is my grant not getting funded? And I think it's maybe you didn't pick the most important question that you could have in front of you. So that's when I was functioning as a department chair and hiring junior faculty, that's what I would challenge them with. Pick your one or two most important questions that you think you can really answer that we want to know the answer to, and just keep working on that. And it'll work out. I don't know, but you'll still get unexpected things when you do that.
Yeah,
right. Because but that's the fun. I think that's the fun of science. It's when things don't turn out the way you thought. I mean, we're all happy when our hypothesis is proven correct, right?
Right.
That always feels satisfying, but really, it's actually more interesting in a way when it's not. I mean, not when it's totally out of control, but when it's something that you can then go and test and figure out.
To be able to test it. Yeah, I was going to ask a little bit about the mentoring aspect. Can you say a little bit more about, besides just work on an interesting question, what do you think are the things that you try to convey as a mentor for young scientists?
Well, I mean, I do think that's very important. I think the other thing that I tell them when they're starting their lab is keep working in the lab yourself for as long as you possibly can. I think that when you're, obviously when you start, you're a small lab, right? But I remind everybody, the reason you're here is probably because you were good in the lab. Right. You were a really good postdoc. You made great data. You're probably going to be the best person in your lab for a while. And also, I think people will learn from you when you're there. Don't just shut yourself in your office and spend all your time reading and writing grants. You have to balance it out, of course.
Yeah.
But I think that, still being hands-on as much as you can. I mean, eventually as your lab gets bigger, you can't do that as much and you get administrative stuff and all that. But as long as you can, I would say that I was still doing stuff at least for the first 8 or 9 years in my lab. Eventually I became other people's technicians for the big experiments. But, my first couple of papers, I had to find somebody else to be the first author so that I could look like I was the last author. Because everybody will, you have your own lab, you want to have your last author. And that actually brings me to my first paper, which we didn't talk about, which I think is another important thing that we've done, which is related, which is the B cells as a target in autoimmunity. Because I know that we were the first people to do that genetically. I think it was only probably the second knockout that was ever crossed onto an autoimmune prone strain.
Oh, is that right?
Well, this is we published it in 1994.
Yeah.
So there was not that many knockouts that had even been made.
Right.
And, I saw right away that the T cell part of the disease was completely gone when I knocked out the B cells. And I realized, okay, these B cells, it's not just antibodies, it's really about antigen presentation.
Yeah.
And, I wrote in the discussion, I think B cells would be a really good target for autoimmunity. We just have to figure out a way to do it.
Yeah.
And that's still, that's had its ups and downs. But at the time that I was doing that, there was no rituximab, there was no anti-CD20 or anything. And then that came along, and it does treat some autoimmunity syndromes, but didn't really do well with lupus. But now these very deep B cell depletions with CAR T cells is working. Yeah. And I think that's excited everybody in the field again. But, again, and I'm still working on that too, but that was the first paper that I published, and I needed to find somebody else to be the first author.
Yeah.
So some of my rotation students got first author papers.
Nice.
Yeah.
So I work with students who are a bit earlier in the process. And so what type of advice do you have for sort of younger students who are interested in science?
How much?
So I work with undergrads.
Undergrads?
Yeah.
I would say have lab experience if you can. Find somebody who's going to teach you. And it doesn't have to be about the PI. I think at that point, undergrads should try to find a graduate student or a postdoc who really wants to take them under their wing, and give them an opportunity to actually do something and think. Because certainly, undergrads, if given that opportunity, and they need a longitudinal experience, I think.
Yes, very true.
But they can contribute to the project. I've had a couple of undergrads who really had some great ideas, actually. Yeah. It doesn't take long for them to get into it. And I think then they figure out whether they have the bug or not, whether or not that getting that idea and testing it or getting those data really turns them on or not.
Awesome.
Yeah.
Very cool.
Yeah.
All right. So I have one final question for you. That is a question that we use sometime on TWIV, another podcast that I'm on. So if you hadn't been a scientist, what would you have done?
Well, I was going to medical school.
Okay.
So I guess the pat answer would have been be a doctor. I eventually, when I did go back to medical school, and I did actually like it. Once I got to the clinical part, it was problem solving, and I liked the people and everything. But I think I could have been involved in maybe government or law or something like that. I don't know. I'm good at arguing.
Awesome.
I think the two things that commonly come up are some sort of artistic thing, like a musician or something related to arguing, because that's what we do.
Oh, really?
Formulate arguments, right?
I definitely, I'm okay at music, but I could never be a musician. I'm not that good.
It's those two things, and also baking.
Oh, baking.
Yes.
Well, which is chemistry, right?
Yeah. No, definitely baking would not be me. No. Cooking maybe, but definitely not baking. Yeah.
Yeah.
Awesome. Well, thank you so much. This is fascinating.
Thanks.
Sure. It was fun chatting with you.
Yeah.
Yes.
Time went by.
That's a special episode of Immune, an immune booster. If you have questions or comments, please send them to immune@microbe.tv. If you enjoy our work, please consider supporting the science shows of Microbe TV. Go to microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today is from the University of Pittsburgh, Mark Shlomchik. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Music on Immune Boosters is by Tatami. Thanks for listening to Immune, the podcast that's infectious.
And you just have to know to recognize it.
Sorry. No, we don't need. Thank you. No, thank you.
Okay, see you tomorrow.
All right, you start. Sorry about that.
That's okay.